All right, so we're picking up right here where we left off, looking at glycolysis. Anyone remember what we said about glycolysis so far? So what is glycolysis? What's kind of the big picture here? Does anyone remember that bit? No, what happened? Okay. So we were saying that glycolysis is the first stage. So if we jump back to this slide, it's the first stage of how we begin breaking down glucose in order to oxidize glucose, in order to take the energy that is stored in that molecule of glucose and liberate that energy so that we can store it in the form of ATP. And then we said ATP was our cell's currency, right? It's what the cells used as energy to fuel their various processes. So we're going to go through each of the stages of how we go from glucose to energy. And the first stage in that process was glycolysis. Now, we also said that glycolysis and the Krebs cycle together were using the mechanism of substrate level phosphorylation, in that they were taking phosphates from another molecule that was present. We said it was a faster, easier way to get that phosphate, add it onto ADP, and then we can make ATP, and again, store up that energy that's liberated from glucose. We also said that there was another mechanism in order to make ATP, which was more sustainable, it was more efficient, it made more ATP, and that was called oxidative phosphorylation. And the stages where that happened were in the electron transport chain, which is sort of the third stage in that process, okay? So let's go ahead and look at what glycolysis is. So we break down a molecule of glucose, and what we get at the very end is 2 pyruvate molecules. Okay, 2 molecules that are called pyruvate. In order to do so, it's a sequence of steps. So it's 10 individual enzyme catalyzed reactions. We're going to look at each of them individually. I don't need you to memorize them. I do need you to know the big picture here, what goes into glycolysis, 1 molecule of glucose, and what comes out at the other end, 2 pyruvate, and what we yield as a result of that process. When I say yield, I mean how much energy, how much ATP are we making when we go ahead and break down that 1 molecule of glucose. Also, I want you to know what we have to put in, okay? So because this is the process of making ATP, we're going to need to put in some ADP in order to phosphorylate them and get ATP on the other side. We're also going to need to put in 2 NAD+, okay? NAD+ are going to be reduced. And if you think back to some of your prior courses and prior knowledge, oxidation is loss of electrons, reduction is gain of electrons. So when we reduce 2 NAD+ molecules, we're actually going to gain 2 electrons here. Let's get that pointer. So we're going to gain 2 electrons here. We can see that happening here. Right, so we input 2 NAD+, which has 2 electrons tagged on. That's what this plus here means. Through the process of glycolysis, we're going to yield 2 electrons from that by reducing it. And those electrons are going to be really important. They're going to be shuttled off into the mitochondria in order to help with oxidative phosphorylation. Okay, so we'll talk about this on the upcoming slides, where these electrons are going, but I want you to make a note here that these electrons are going to the mitochondria, and they're going to be important for that third stage of the oxidative phosphorylation, and that's going to be happening using the electron transport chain, okay? So we input 2 NAD+, we get 2 electrons here that go somewhere else to do something that's really important. We also input 2 ADP along with 2 phosphates. Remember, this is an example of substrate level phosphorylation. So we're going to grab these phosphates from other molecules, and we're going to add them onto the ADP, and we're going to yield 2 ATP. Okay, as a part of this process, there is absolutely no oxygen required, and there is no carbon dioxide produced. Okay, this is what I mean by this being a fast, cheap, easy way to make ATP. Where this is going to happen in the cytosol of the cell. So if we think about a cell, right, it's got the floating sort of fluid environment where everything is suspended. Then we have some organelles in the cell. We've got things like the nucleus. We've got things like the mitochondria. We've got things like the endoplasmic reticulum, whether it be rough or smooth. And so where this is happening is just free floating in the cytosol, not necessarily in any one organelle, okay? If we were to focus a little bit more at the equation, so the overall big picture of what's happening with glycolysis, we input 1 molecule of glucose, we input 2 NAD+, which again is sort of carrying 2 electrons with it. We input 2 ADP, and we grab 2 phosphates from other molecules in the cytosol. And what we get on the other end are 2-pyruvate. So this is our most important yield from glycolysis, 2-pyruvate. We're going to yield 2 NADH and the 2 electrons. Okay, so we reduced NAD+ to now harvest those 2 electrons to go somewhere else. And then we're going to yield the 2 ATP. Okay, everyone with me so far? Any questions on this sort of big picture of what's happening? Okay. And I'm hoping I'm sharing the correct screen. So can you guys see my cursor? Can you guys see my cursor moving here? I just want to make sure I'm sure. Okay, perfect. Thank you. All right, so that's glycolysis. That's what we've got so far. Let's go ahead and look at the individual enzyme catalyzed steps. So this is a very complicated process. If you happen to go to med school or graduate school, you probably may be required to memorize each of the enzymes, each of the products, each of the substrates here, and each of the subsequent steps. But for this purpose, for the purpose of this class, you aren't necessarily required to remember the individual steps, but I do want you to know what's happening specifically as it pertains to ATP. Okay. So essentially, we've got glucose here to start. That's kind of the most important step. And then the end product is also the most important. We get 2-pyruvate. So the first few steps, the first 3 enzyme catalyzed steps of the reaction, where we go from glucose to fructose 1,6-bisphosphate, this is called the energy investment phase. Now we're going to have to put some ATP in here and invest it on the front end, okay? And so this is why, although we yield 4 ATP total, we're only yielding 2 net ATP because we had to put 2 in to begin with. So the energy investment phase is the first 3 steps of glycolysis. We had to put 2 ATP molecules in, 1 to undergo the first reaction, 1 to undergo the second reaction. And so it's going to cost us 2 ATP up front just to get glycolysis started. At around the 3rd step here, what ends up happening is that we have 2 G3P, 2 glyceraldehyde 3-phosphate molecules. And so from this stage onward, we have 2 molecules yielded per glucose that we have. So for 1 glucose, we have 2 G3P, we have 2 1,3-bisphosphoglycerate, and then 2 of each of the other substrates. This is how we end up yielding 2-pyruvate. all right? Because this one fructose 1,6-bisphosphate ends up splitting into two so that we end up having two yield for the rest of the equation or the rest of the reactions. So we end up seeing 2 ATP made here and 2 ATP made here. And so as a total yield, we end up seeing 4 ATP in the energy harvesting phase, which is a total of 2 net ATP. Okay, again, I don't need you to memorize anything here, but I want you to understand why we end up seeing 4 ATP, because of the fact that we now, at this stage in the reaction, we cleave fructose 1,6-bisphosphate into 2 separate G3P molecules. So the rest of the steps are going to happen twice, and so we yield 4 ATP. Okay, here's another representation of how all of these steps happen. Again, we invest 1 ATP, we invest another. From this stage onward, the reaction is doubling. So now we're going to yield 2 ATP here and 2 ATP here, and our final end product will be 2 molecules of pyruvate for every single glucose that we break down. Okay. Let's move on to talk about what's happening in the linking step. So what happens to that pyruvate? Let's kind of pick up from there. The pyruvate is going to then be broken down. This is happening in the mitochondrial matrix. And it's going to go from pyruvate with the help of the enzyme pyruvate dehydrogenase. We're going to yield acetyl-CoA, which is this molecule here. Right, it's an acetyl group with CoA attached, and then we're also going to yield some byproducts. So we're going to yield some carbon dioxide. We're going to yield some more electrons by reducing more NAD+ to give us NADH, and then the free-floating electron. Okay, so this is the second stage that we're looking at. This is not usually considered a stage of its own because it's sort of a linking intermediate step. So if you think back to the stages that we talked about earlier, it wasn't listed there, but it is an intermediary where we convert pyruvate to acetyl-CoA, and then acetyl-CoA will go on to the third step or third stage, which is the Krebs cycle. All right. What I do want to point out and emphasize here is that we begin to make some byproducts. So in glycolysis, we made no carbon dioxide. Here, we begin to make some of these byproducts, such as carbon dioxide, and that carbon dioxide is what we exhale. This is the cost of breaking down glucose. This is why we breathe out carbon dioxide, because it's the byproduct from our cells after they break down glucose, liberate energy, and then release carbon dioxide as a waste. Oh, sorry. I did also want to mention here that we have to invest. What are the things that we have to invest? We're investing the pyruvate. So 2 pyruvate, remember, glycolysis yielded 2 pyruvate. But for every single pyruvate, this reaction is going to happen. And so for 1 molecule of glucose, we're having 2 acetyl-CoA, 2 CO2, and then 2 NADH+ with 2 electrons as well. Last thing to note here is that the electrons are going to be going to the mitochondria to be a part of oxidative phosphorylation. So keep bearing that in mind and keep making a note of that on your slide, that the electrons that we're making, they're going to be doing something else in the mitochondria. So we don't want to ignore them here. We want to make a note that they're going to be going towards oxidative phosphorylation. Okay, let's talk about the Krebs cycle. So the Krebs cycle, again, another very complicated series or a sequence of enzymatic reactions. This one happens to be a cyclical series of reactions in that our substrate and our product is the exact same. So we put those acetyl-CoA that we just made in that linking step into this cyclical set of reactions. And that is going to yield us more electrons. We're going to reduce more NAD+ to give us more electrons, specifically 3 electrons per acetyl-CoA. We're now going to yield another type of electron. So we're also going to reduce FAD to FADH2. This is just another way for us to liberate more electrons, okay? We're also going to invest some water, so 3H2O, and then we're going to yield more carbon dioxide as a waste product here. And then finally, probably the most important yield is going to be one ATP molecule, and we had to invest one ADP and one phosphate, okay? So if we look at our reaction at the bottom here, we're putting in the acetyl-CoA. Remember that for one molecule of glucose, we had 2 pyruvate, so that means we have 2 acetyl-CoA. All right, we're going to invest 3 NAD+. We're going to invest 1 FAD. Both of these are just ways for us to get electrons liberated by reduction. We're going to invest ADP and phosphate. That's going to be our substrates for making that ATP. And then we're going to invest some water as well. And then on the product end, we're going to end up seeing 2 carbon dioxide, 3 NADH carrying 3 electrons, and then FADH2 also carrying 2 electrons here, our ATP, 1 ATP molecule per acetyl-CoA. And then we're going to end up having CoA as a byproduct as well, okay? All right, here's what the Krebs cycle looks like. Just bearing in mind here, because this is a cycle, we put in acetyl-CoA and we end up, again, yielding acetyl-CoA. But along the way, we get the various additional products that we've mentioned. So the electrons, the carbon dioxide, the additional electrons via FADH, the water, and then we get back that acetyl-CoA that we begun with, okay? Again, I don't need you to memorize each of the enzymes, each of the substrates, each of the steps, but I do want you to know that we put in acetyl-CoA and we get back acetyl-CoA along with the additional yield products that we have mentioned. Okay, I'm going to pause here again and see if there are any questions that pop up, and I want to get into the third and final and probably most important step or stage, which is the oxidative phosphorylation. Okay, now contrasting to what we have described so far, glycolysis, the linking step, the Krebs cycle, they only yield us a net of 4 ATP. There's 2 net ATP coming from glycolysis, and then there is 1 ATP coming from the linking step, 1 ATP coming from the Krebs cycle. So together, as we have begun breaking down one molecule of glucose, we've only really gotten 4 ATP molecules. That's not a whole lot, right? It was easy, it was pretty fast, and it's happening out in the cytosol, so to speak, but it's not a lot of ATP. It's not enough to sustain our cells and all the work that our cells have to do. So this is where oxidative phosphorylation comes into play. It's a way for us to make a lot more ATP. This is where the bulk of ATP is made. It's called the electron transport chain. So if you remember all of the electrons that I've been mentioning, that we've been building up, that we've been gathering, that we've been making, they are going to be a really important part of how we make this bulk of ATP via a mechanism called the electron transport chain. And the big picture of what's happening here is we're going to move electrons down their electron transport chain, and we're going to use that energy. The movement of those electrons is going to yield the energy that's going to make a lot more ATP. We're going to need to have oxygen for this step, right? Hence the oxidative part of this process. We're going to need to invest oxygen. In fact, fun fact, this is the only reason that we breathe in oxygen, in order to drive oxidative phosphorylation, in order to make a whole lot of ATP, okay? This is the sole reason that we breathe in and out, you know, a couple of minutes or a couple of seconds, a couple of times each minute is what I'm trying to say. every single day. This is why we need to have oxygen. This is why we have to oxygenate our tissues and our cells and our entire body in order for that oxygen to be used to drive oxidative phosphorylation to make ATP, okay? So let's talk about exactly what's happening with the electron transport chain. So we have the mitochondria. This is where this is taking place. We have two layers to the mitochondria. There's an inner mitochondrial membrane, and there is an outer mitochondrial membrane. And across that inner mitochondrial membrane is where we have these electron transporters. So the electron transporters are driving electrons that are undergoing an oxidation reduction reaction, going from high to low energy. So again, remind ourselves here that oxidation is loss, reduction is gain. So while we were undergoing reduction reactions to make these electrons in glycolysis and in the Krebs cycle, here we're going to undergo the opposite reaction. We're going to take those electrons, we're going to oxidize them, right? They're going to now lose that. We're going to lose those electrons. We're going to take that energy and we're going to use it to make ATP. Okay. Now, oxygen. What does oxygen serve in all of this? It's just going to accept those electrons. So as it accepts those electrons, the energy from that transfer, the energy from that movement is what we use to make our ATP. And as I keep stressing here, this is going to make a lot more ATP, which is going to sustain our cells, our tissues, our body.